You know, when I was asked to do this, I felt very, very honored, of course, to give a talk in Lindon's honor. Then, of course, the dreaded moment comes when they ask, what are you going to talk about? My goodness, you have one of those, okay, now what the colloquial term for coitus am I going to do? And so what happened is I began thinking of the various and sundry types of work that Lindon and I sort of did in common. One of the biggest and most important works that Lindon contributed to is a model of sibling effects in man for this. So I thought, oh, good, here we go. You know, we could have talk about this and sibling interactions, okay? Another thing that Lindon ended up talking about was generalized effects of assortative mating, which I also had a lot of interest in. So we have something like this, and I thought, well, let's not go there. So as I was doing this, it turned out I was also working on a project with a graduate student, Amanda Wills, and at the time, I was putting everything together, trying to do this sibling effects assortment, and Amanda was doing this, and came up with something that, instead of being sort of the jocular, showing all these wonderful pictures, is something that I really think is my biggest tribute to Lindon, and that is to present something original, new science, okay? On the aforementioned topics. Why friends? Mainly because we were working on data on smoking in adolescents and twins and their friends. The basic observations, of course, we already know. Identical twins have higher correlations than fraternal twins, okay? We know that identical twins have more friends in common than fraternal twins. That's been demonstrated. The interesting thing, especially for these particular data and for some others, is that if you look at the friends of identical twins, they resemble each other more than the friends of fraternal twins. Let's forget about that. Those are twin correlations. You've already seen them. Here are Ad Health data, where the twins had to say of your 3 best friends, how many smoke, okay? And you will notice, if you look and compare the column for MZ twins and DZ twins, same sex in this case, for waves 1 and 2, you can see that the correlation for the friends of identical twins exceeds that for fraternal twins, okay? So here's the question, what's going on? Okay, well, to figure out what's going on, one has to at least start initially to see a model about what is going on. And we have identified 4 different processes that can contribute to the correlation. The data that we want to take a look at is to model the processes for, whoa, that's a pencil. We don't, I thought there was some type of special Yoda Star Wars thing that was going to show up. Pardon? Use the mouse. Oh, that works. Good. Modern technology, man. Okay, but we have twin 1, twin 2, friend 1, friend 2, and what we would like to do is take the processes that are going to generate a covariance matrix among these 4 variables. The first process is something called, that we're familiar with, but only in part, namely unequal twin environments, where the correlation between friends for 1 and friend for 2 is a parameter that's a function of zygosity, okay? Why this? Well, remember, we are looking at adolescence. We have a large amount of data on adults and the similarity among friends for identical twins as kids growing up and their effect on adult behavior. This is on adolescent smoking. We have to at least keep some type of open mind that perhaps just because Harry and Hermine look identical, they tend to get invited to the same types of parties and so on and so forth more often, and that induces a correlation between the friends. Notice that in this model, this is the unequal twins environment that most people think of, where we have a correlation between friends 1 and friends 2 here, but that also influences the twin correlation, okay? That is not necessary in this particular model. Okay, may do so, may not. So unequal twin environments is the first one. Second thing are background environments. People in the assortment business call this social homogamy, but all we have to do is recognize that there are such things, such as geographical variations in smoking. The AdHealth sample is from America, and there are different regional differences in smoking. Within regions, of course, you will have differences in such things as family background. Working class have a tendency to puff more than folks that are a little bit more educated and all of that. And if that happens with twins, it can also happen certainly with their friends. So we have the background environments as the second one. The third major process is something called homophily. This is not a term that we use in behavioral genetics very much, but it is a very a term that is used in particularly sociology and social studies. And it just means that people that have similar interests, talents, blah, 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 tend to congregate together, the birds of a feather model. And here, again, we can use this using the assortment types of parameters, or the methods that we use for assortment. And I'll denote this parameter by H for homophily, going there. And that will induce a correlation, certainly between twin and friends. By the way, there's going to be other stuff I should have mentioned up here that will correlate twin 1 and twin 2. I'm assuming that that's in the model, and just don't show it to make sure it's uncluttered. The last possible influence is peer influence, where the actual behavior of one person influences the behavior of the other. It could be twin 1, twin 2, and again, we have to make note that that could be a different effect than it could be between twin 1 and twin 1's friend, or twin 1 and twin 2's friend, and so on and so forth. So we end up having a complicated diagram like this. It looks like Sitting Bull's battle plan for the little bighorn. But it does have a sufficient number, it does have parameters in there for it. Okay, great. So these are the processes. Now people would say, well, fine, simple. All we have to do is go back to our twin workshop and plug it all in. And that doesn't work. Here's where we have to start getting original. The current state of the art in terms of modeling these different processes is actually developed by David Folker and Kay Phillips. That's the one that they use. However, it gives stupid answers. If we were to apply this, for example, to height, go back and put all those, all the processes together, use the typical procedure, put it in for height, then peers that end up assorting for height will have the physical effect of making tall folks grow taller and shorter folks shrink, which is not really a particularly good idea. So what we have to do is go back to the drawing board and come up with a model that will end up doing it. The solution is really simple, okay? The first thing you have to recognize is that before peers can influence one another, they have to form peers groups to begin with, okay? So the process that we used to model this is to start out with the formation of groups, okay? Point 1. And point 2, the second major idea, is that these processes are independent and additive, okay? I'm not going to go through all the algebra, yuck, real boring, for all that. But the two-step process is rather simple. First, peer groups form. Second, then they can influence one another. Okay? And this gets us out of the whole big, this along with the assumption of additivity, gets us out of the whole big problem of people shrinking and growing based on the friends that they happen to associate with. Okay? So here we go. This is actually, again, what are we trying to explain? It's the covariance among these four variables. And what is the formation going to happen? Well, the very first thing that's happening is that the twin pairs are there. Okay, so we have antecedent twin correlations, covariances, and so on and so forth. Then we add in the effects of unequal environments. Okay, so we take this matrix, for example, go up in it, and put in an FZ, one for identical twins, another one for fraternal twins, into that matrix. Continuously go down and add in the effect of shared background, and finally, the effect of homophily for it. After that, now, we have peer groups formed. Okay? So what we do now is the covariance matrix after peer formation and peer influence equals the stuff we had before plus the effect of peer influence. And how do we model that? Well, we go back to the seminal paper that Lindon wrote, and I just use this in path terms. There are slight differences between the path model and Lindon's, but they're really inconsequential for all of that for it. Okay? All this is good. Guess what? I never believed this. The actual thing is identified, honestly. I shouldn't say it, but it was almost like a commercial for X-Lax when this thing popped up on the screen. Whoa! You know, that did it, despite all those god-awful U's and W's and V's and X's and so on and so forth in there and everything else. The model is identified, and guess why? Just as Lindon had shown earlier, it's identified because it predicts different variances for identical twins, fraternal twins, the friends of identical twins, the friends of fraternal twins, and so on and so forth. So one of the nice things about this is we have a great new model, well, I wouldn't say great, but at least we have a model that can help us to sort this out. Okay, here's the bad news. Okay, in order to actually estimate everything, we have to have the same phenotypes measured in twins and in their friends. Data that I showed you before, whoopsie. Okay, we have data on smoking in the twins, but what about their friends? We don't have the same data on that, okay? So this model is very good theoretically, can help you out and all that, but in a scientific sense, I also think this is a good tribute to Lindon. It's not always that when you're working on a problem that you know the exact answer, but often it tells you what type of data to get to really get the answer. And so we've learned something, I think, important through this exercise, namely that if we do want to begin looking at such things as peer group formations and influences early on, then we can end up doing this, but we have to gather the same data, okay? Conclusions, okay? The basic conclusion is, again, that our data can, using genetically informative data, can end up helping us to get a grasp on the nature of correlations among peers. Also, something I didn't go into, but is relatively obvious, we can indeed reject simple models. We could reject the simple model, for example, of peer influence as being the sole and only thing by looking at twin variances, for example, okay? If they don't differ very much, we know that that is not the real big major player in this whole business. So we can reject some simple models, but again, to be fully informative, we really need to start collecting somewhat different data on this. Okay? Thank you very much.
Well, I feel it is a great honor to be one of the speakers today, and also extremely privileged to have experienced Lindon as part of my environment for the last 20 to 25 years. And maybe the title of my talk is rather boring compared to lots of the other speakers here. I'm supposed to talk about extended twin models and about adolescent behavioral development. And so to lighten it up a bit, I decided to try to tell a bit of a story in pictures from way back to about now. And also, I'm very visual, so I like to, path diagrams are sort of a main focus in trying to understand models. So I'll use that to try to build a story. I met Lindon back in 1987 at a workshop which luckily sort of was the start of a series of workshops. We had two in Belgium in 87 and 89. And clearly they have had an enormous influence on where my life has ended up. But I am extremely grateful for that. So this, I don't know if you can read it up here. This is actually Nick's quote on the picture that you've now seen a couple of times. According to my twin model, everything can be figured out except how to live. One of the things that, or one of the graphs, I think, that sort of has influenced me, and I think is sort of a basis of talking to students in the field, is this theory model data graph that I think the earliest version was in this paper, The Utility of Twins, back in 1982. And so the key piece here is that once you formulate a problem, the important part is to try to translate that into a model that is testable with real data to then go back and see whether indeed the model fits the data and the theory is supported or not. And some of these other basic issues that were set out in an early paper by him is that it's important to question before you start, does the approach that I'm going to take, is it useful and will it allow me to really test realistic hypotheses? Does the proposed constellation of relatives really allow the discrimination of the alternative hypotheses? And am I using the right method of analysis to really make best use of the available data? So he really, I think, stressed this to me, but also to many of talks in which students or other colleagues sort of tried to talk about their work is what's the question behind that you're trying to answer, and really is it properly formulated in terms of a model? I mean, this is a theme that has come up in several talks already, but then of course take that to the next step and collect the data to see if the model indeed fits the data. Now, whenever we come up with a model, even the basic simple twin model testing the role of genes and environment, there's always a ton of assumptions that we make, and we're not always very explicit about it. But going back to an early paper of Lindon, I mean, he had about 8 assumptions in here, and I think it's important to recognize both the strengths and the limitations of our model, and when formulating the model, keep that in mind. Below here is an early picture of one of the early workshops with David Fulker teaching matrix algebra, which was a real experience. It's lovely to think back of. Lindon is also, of course, key in thinking beyond or realizing that just a basic twin model would not answer all our questions, and we might need to go beyond to be able to test a lot of the assumptions that we have to make in the twin model. And so this is sort of the first introduction of dealing with genotype, environment, correlation, interaction, as well as going to the extended model with the genetic and cultural transmission at the bottom here to extend the model beyond twins to be able to evaluate the consistency of the results across various designs, and to take in more sources of variation and covariation and build a more realistic model. So what I'm going to do here is sort of walk through a couple of graphs that I found throughout Lindon's papers that sort of really try to depict a different kind of model for the influences of parents on children, or broadly, not only twins, but also singletons. So this one is from an early paper, Model Fitting Approaches, which I mean, it's about a 40-page paper that sort of lays the groundwork of pretty much most of the work that we do here. So I highly recommend, especially the new graduate students, to go back and read some of these old papers. So here we have the parents influencing the environment of the children, so a form of environmental transmission, but then there is also potentially environmental transmission from one twin onto the other twin. So we have some sibling environment as well, and this is all happening in the context of assortment between the parents. This version here is a P2P transmission, so supposedly from the phenotype of the parents to the phenotype of the children. I'm not quite sure about this graph here. Some have gotten some names here. I think we need to come up with a nice name for this one. But this is direct transmission from phenotype to phenotype, but in the presence, of course, of genetic correlation between parents and offspring. Of course, again, these models make very strong predictions, and so the influence supposedly would go from the parents to the kids, but for everyone who has kids, I think they influence you in ways that you could not imagine before having them. This model is probably the one that most people are familiar with, because it's the one that we most often fit when we have data on twins or children and parents, and it includes both the genetic transmission from parents to offspring, as well as the cultural transmission, here modeled from the phenotype of the parent to the environment of the child, again in the context of assortment between the parents, again based on the phenotype of the parent. The combined presence of genetic and cultural transmission then leads to potential genotype-environment covariance, which is then assumed to be the same across generations. Now, of course, one can model assortment in different ways, and here it is modeled as assortment for a latent environmental variable, which is then transmitted directly onto the environment of the offspring, so that we call that the E to E transmission. Or a slightly different version of this, where it's only part of the environment for which there is assortment, but it's perfect assortment that is then completely transmitted onto the offspring. This is another typical graph of the social homogamy model, where again we have the correlation between the partners modeled in terms of their shared environment that contributes to the shared environment in the offspring, allowing also for additional influences to contribute to the environment in the offspring that's not coming from the parents. And then finally, this one here, I think this is one of Andrew's, even though he's not directly mentioned here, but it's a very complicated graph, and I haven't quite figured it out yet, but I know it's supposed to be about the mixed homogamy, so a combination of both phenotypic assortment as well as social homogamy, as well as allowing for errors of measurement and all possible other things. So Andrew, if you can explain this to me one day, that would be lovely. And beyond sort of all these different models, of course, it was Lindon who came up with the idea of going beyond even the twins and the parents, and including other relatives to try to see whether or not the results that one obtains can be generalized or are consistent across all these other relatives. And so by using that basic model and taking the expectations from that, adding other relatives that are more remote, could then build into the model and all analyzed together. Now, before, even though it was hard to find some of these graphs, and for me, graphs have always been sort of the key to try to understand models, but clearly Lindon has quite a few more IQ points, and he actually worked out all the expectations way back in 1976 in this lovely paper on cultural transmission on continuous variation, and also spelled out all the potential assumptions that these models make, and he can do it in his sleep. It's just second nature to him. So after sort of coming up with all these great models to test alternative mechanisms of cultural and genetic transmission, he of course had to collect some data to test some of these models, and so the design of the Virginia 30,000 came about, and Nick has already alluded to that, with twins in the middle, their parents, their spouses, their siblings, their children, which led to a variety of papers, and you've seen the graph, which I think was called Stealth for a while. I don't know if this is the particular graph that was termed Stealth because the picture looked like a stealth bomber. There have been a variety of names, and I've worked on these data as well, and came up with a slightly different graph. And because it didn't look anything like Stealth, I called it ET. Could refer to the figure ET, but it's really just extended kinships, twin kinships. And the only one difference here between these graphs is the fact that I have the double-headed arrows on the latent variables, and we've argued about this many times. I think there's great value in having a simple model and sort of focusing on just the important parts of it. However, I tend to put in the arrows and make the model not only aesthetically beautiful, if possible, but also mathematically correct, so you can derive the expectations and make sure that you don't forget about any of the assumptions that you make. So I think we've taken... oh, sorry, Lindon. This is... So when Lindon programmed this amazing stealth model in Fortran, we've taken it from there and put it into first in classical MX and now in OpenMX. From dealing with originally just the correlations, we now can also fit to raw data. It is set up to do bivariate and multivariate analyses, although in practice that's still quite tricky. In addition to fitting phenotypic assortment, there's also Matt Keller's work turning the model into one that also allows for social homogamy. We can easily moderate the means by age or any other covariate, and hoping to move into the direction of also moderating the variance components and deal with the one thing that the model so far doesn't deal with yet is genotype by age interaction, but I'm hoping that we can make some progress towards doing that as well. So here is actually the version of Matt Keller. I don't know if this is cultural transmission from, but somehow I mean, whether or not they fall asleep, they seem to know exactly when what question to answer when they do wake up. Now, so talking about pedigrees and going back to Andrew's talk, he said, well, we should sort of have Lindon's pedigree. So this is my attempt at Lindon's pedigree. Now, for those, I mean, there's lots more people in the audience that he's influenced that are not on these pictures. These are actually just the pictures of the speakers from today. And I've taken some liberty with this pedigree. Obviously, you have Lindon up there. I had to have David in there, and I don't know if that's a sort of gay marriage or whatever. It doesn't really matter. But and then they have twin offspring. That's Nick and Andrew, the two students of Lindon, a bunch of other people. Now, the generations, of course, sort of flow into one another, and the fact that I have them all connected with lots of lines from here or there's lots of influences going back and forth between all these folks. And then the lines at the bottom sort of represent the further influence to the rest of the BG and the wider community. And I think Lindon must have more than been about 1,000 students and counting through workshops and presentations back home. And I thought I'd put this picture at the bottom in here, just looking at the people standing up. I would like to know what the average IQ is, but I'm sure it's a bit higher than mine. And now there's something entirely different. I was supposed to talk about not just extended twin models, but also the adolescent development, behavioral development project. So I thought, well, so we're talking about age and development and whether or not genes, potentially different genes play a role at different times during the lifespan, or that their effects are different. So I thought, well, let's have a look at Lindon over the years. So these are pictures, the dates at the bottom are the years in which the pictures were taken from back in 91 to just last year. And so Lindon has always been a key person in trying to look at developmental questions. And one of the early developmental models is a fairly simple model that suggests that most of the environmental influences are time-specific, but that there is some transmission of genetic effects from one time point to the next. And a slightly more complex version of this model sort of also allows, in addition to the transmissions and the innovations, also for some common influences that remain throughout development. And then I found this model here, which I think has not been used all that much. It's sort of twin data, but over time, with time 0 and time 1 up there, but where there is the possibility also combining that with direct influence from one time point on the next, as well as from twin 1 on twin 2, so some version of sibling interaction and causation all put together. And so Lindon, I mean, I think has done a great job in formulating models, but also, and can teach about any topic that has come up so far, but also is very good in explaining the basic concepts in how we actually then test these models and do the model fitting. So here we have a couple of pictures from early talks on power and hypothesis testing over the years. And when before going to actually collect data, he then also often does simulations to make sure to have some idea of what kind of answer one might expect given certain situations. And again, he can do that in his sleep as well. And of course, the other thing that's critical in doing any of this work is power. So, so here I'm going to focus a little bit on the VTS-ABD, the Virginia Twin Study of Adolescent Behavioral Development, actually started by John Hewitt, but then taken over by Lindon when John moved to Colorado. And so where you can really see that how you design the study then has enormous implications on what kinds of questions you can answer with that particular study. So having twins obviously allows standard ACE modeling, adding in the parents to be able to elucidate genetic and cultural transmission and assortment, adding both male, female, and opposite-sex twins to test heterogeneity across gender, including longitudinal data to fit all these developmental models, adding multiple raters, having different kinds of measures, both dimensional and categorical, to do various multivariate kind of approaches, factor analysis, IRT, latent class analysis. I mean, Lindon has really, sort of set out the ways for all these different sort of aspects, including environmental to do environmental indices to test for G by E interaction or in the presence of GE correlation, dealing with onset and puberty and how that plays in, and even now sort of having some genotypes. So he's now thinking through clearly, how we will use those genotypes in our modeling framework to answer even more questions. And so through this project, he's worked with a lot of people, and this is a graph from, it's called Visual Explorer, where you can put your name in, and it then brings up all the people that you have co-authored papers with. And so the ones closer to you are the ones, I guess, you have co-authored more papers with, and so it's a whole network of things. Unfortunately, it doesn't have pictures of everyone just yet. We need to work on that. And on the right here is just the sort of overview of his papers, the ones at least that are in Web of Science. You see who he's primarily worked with, but he's worked with a lot of people, and these only include the people he has at least 2 publications with. Another way, of course, of looking at his impact is looking at how often people cite his work. And so this is another graph here with a citation graph where you see a lot more of you guys popping up as obviously he's published quite a bit and quite often in behavior genetics. About 61 papers have been published with Lindon in behavior genetics. So again, I think he's had an enormous impact on the field through this, partly through the workshops, where if you even look at all the people that have taught there, they're all sort of interconnected, and it's been a great sort of family, so to speak, to grow up in and do this kind of work. And so to go back to the theory model data graph that we started with in the beginning, this is actually from the most recent workshop where Lindon gave one of the final talks. And so if you can write the model, you can fit it, at least if you can get the data. Now, chances are that your real questions don't fit into the basic framework, so be creative, imagine. And thinking of the future, I guess it's still an open question as to how the modeling sort of integrates with the gene hunting business. And this is back to Mike Kearsey. He sort of had the same quote. This is, again, in one of those classical papers that discusses one of Lindon's first papers. It's actually a response to comments by, I think, Dr. Vetta of John Jinks saying, I could quote many examples to show that the number of genes found is proportional to the patients and the effort which the experimenter is willing to put into their detection. Now, when Lindon says this in his accent, it sounds way better, and it's way more funny than I can ever do it. But anyway, it's been a great ride so far, and I'm looking forward to continuing to work with you. And when I sort of had to come up with a couple of adjectives to describe Lindon, generous is certainly the one that comes to mind most often. I mean, he's been extremely generous both with his time, his data, his intellect. He's a true genius, and you can read the others. And I'm looking forward to having a beer and celebrate further throughout the rest of the conference. Thank you.
Thanks for inviting me to be here. It's tedious for PhD students and graduate students to hear, but what Hermine said is true. You should all go back and read Lindon's papers. I was invited here to give a talk on G by E interaction and G by E covariance, which I'll do. I was surprised to note that Dorrit is my mother. I always view you as my older, wiser sister. Anyway, my background is the following. In the 1980s, I was studying developmental psychology at the University of Amsterdam, and genetic hypotheses at that time were considered incoherent, simply because the interaction and interplay of genes and environment was such that any genetic hypothesis would be rendered incoherent. Strangely, the University of Amsterdam is a university with a slightly leftist slant, and strangely, at the same time, environmental hypotheses were viewed as perfectly okay. Now, in the mid-90s, by the time I had finished my PhD, twin and family studies had become, and still were, making a huge contribution to differential psychology, and it's the study of individual differences. And this work was largely embraced by my colleagues at the University of Amsterdam and by everybody else. And this shift in perspective is largely due, I think, to the work of Lindon Eaves and his colleagues. Specifically, in the 1970s, as people have already mentioned, Lindon and his colleagues produced a series of papers which provided the theoretical, conceptual, and the statistical basis for genetic covariance structure modeling. These papers, in combination with software developments, namely, specifically, the development of the MX program, the MX missile, as Nick used to call it, had a huge influence on different psychology and would keep a generation of researchers, including myself, busy for the subsequent decades. So what happened to the interplay of the genetic and the environmental, rendering genetic hypotheses incoherent? I suppose it must have dawned on my developmental colleagues that the study of individual differences, developmental behavior genetics, is not the study of developmental behavioral ontogeny, two different things, and their confusion of the two has always been a great source of controversy. What happened to interaction? Within the field of developmental psychology, it remained a largely theoretical concept, and within the field of behavior genetics, it was treated appropriately, properly, and clearly by Lindon Eaves, notably in this paper, which is one of the papers that all the graduate students should study. Now, in Lindon's treatment, the statistical view of gene-by-E interaction is one of genetic control of sensitivity to the environment. This in turn leads to a view of G-by-E correlation, G-by-E interaction, as one giving rise to heteroscedasticity. And this is just a caricature. So this shows you the genetic score, if you can get at it, against the phenotypic score, and you'll note that there's an increase in dispersion, which represents the increase in sensitivity of the genotype to the influences of the environment, as manifesting in the phenotypic score. Now, this is just an example. Increasing dispersion with increasing score is not the only possibility of systematic relationship. But the question is, why should the relationship be systematic, right? And this is discussed in detail by Lindon Eaves. In some papers, he will say, based on the study of experimental organisms, that the relationship may well be systematic. And in other papers, he says that the relationship is probably not systematic. There's a reason for this, which I'll return to in a second. But in the 77 paper, from the point of view of the animal studies, Lindon says that often a substantial part of the G-by-E variation is due to the effects of genes which are associated with the genes which contribute to the differences between genotypes averaged over environments. So I suppose this is interpretable as a systematic effect. Not necessarily linear, of course. He also notes he has two more substantive interpretations, which I liked. The first one concerns intelligence. He says it's not beyond the bounds of possibility that a genetical system could have evolved in which the genes increase intelligence, which increase intelligence are associated with the genes that promote stability. Very nice, interesting, and thought-provoking interpretation, and it links to something I'm interested in, that is ability differentiation, the notion that general intelligence is a stronger source of individual differences, resulting in higher correlations at the lower end of the IQ distribution than it is at the higher end of the distribution. And this is an interpretation thereof, or explanation thereof. He has another quote, which I'll skip because I can't imagine, there's no narrative that presents itself to me. And that is individuals who, on average, manifest high scores may find themselves in a more variable environment, in more variable environments because there is no consistent policy towards the treatment of such individuals. So that's what?
You say rot.
What? No, I didn't say rot. I said, so what was rot? Okay. I thought maybe highly aggressive people, or just okay. I made a huge impression on me. Okay, well, one of the, so when we're talking about systematic G-by interaction, one of the most, the simplest tests, which was studied extensively by Nick Martin under the supervision of, I imagine, of Lindon Eaves, and that is the famous Jinks-Fulker test, which involves the polynomial regression of the absolute MZA differences on the MZA sums, where MZ stands for monozygotic and A stands for apart. This is a very simple test. Can be carried out using MZs grown up together, but that is not nice because it may confine C-by-E and G-by-E interaction. And that's bad because Lindon views C and E as causally distinct. There's a paper by Dr. Carey that made a huge impression on me, but I don't think it made an impression on anybody else, about precisely this, whether C and E should be viewed as being causally distinct.
Now
this test is, as I mentioned, I like it because it's simple. It's easy to understand. It's easy to conduct statistically in SPSS, whatever you use. But it comes with the disadvantage that it is skill dependent. And I'll return to skill dependency in a second. But before doing so, I'd like to mention two generalizations of this test, which Lindon developed in 1972 and 1976. One being the multivariate version of this test, which I found very, very interesting, and the other being the introduction of age as a moderator variable. Nick already mentioned this paper. This paper, the genotype by environmental paper, the genotype by age interaction paper, set the stage for conditioning on an external variable, and that resulted in important work by Daniel Dick, and the Sean Purcell work, and that resulted in very many papers in which people would take an external variable such as age and then introduce it in the twin model as a moderator. And this paper in 1976 presages, how do you pronounce that? Presages that type of model. The multivariate test is of interest to me, again, because I'm interested in ability differentiation. If there's G by E at the level of general intelligence, and if you can interpret that as G by E, as a form of ability differentiation, and if general intelligence underpins all the various cognitive ability tests, then you'd expect that effect to be manifest at the subtest level. So it is important to have a statistical test of the minimum number of dimensions required to represent these interactions and to allow their structure to be investigated. General intelligence would be an ideal test case, I think. Now, I already mentioned the problem of skill dependency. So what of this long dark shadow that apparently spoils the fun with respect to the Jinks and Fokker test? Firstly, I should mention that Lindon discussed this extensively in the 1977 paper, and it's very interesting. Close reading is always good, but in the case of Lindon, it's very, very good, because you can see, I think, how he grapples with this, because it's not a simple problem. Firstly, Lindon and his colleagues demonstrate it very nicely. They take 74 neuroticism items, and they fit these using an IRT program, which at the time, I think, was pretty sensational in its own right, computationally. They calculate the raw scores and the raw transform scores using an arc-sine transformation, I think. And each phenotype produces a different form of G-by interaction using I think the Jinks and Fokker test. So this is a demonstration, and an appropriate warning is issued that this is the case. Lindon has also more recently demonstrated the same thing using measured genotypes and measured environments in a simulation setting in the 2006 paper, showing that using a dependent or a dichotomized dependent variable and logistic regression doesn't help. So the problem is still there. Finally, there's one paper I'd like to mention. It's not about G-by interaction, but I really enjoyed reading it. And that is the same problem occurs when you use mixture modeling to try to detect the presence of major genes in otherwise continuous data. And you have the same problem. Bad scaling will produce components which are a function of bad scaling, not a function of any major genes. So what about all this? I mean, we understand that there is such a thing as skill dependency, but what should we do about it? And that is where I think the 77 paper is very interesting, because Lindon emerges in my mind as a realist and a pragmatist. It's not correct, huh? Okay. Well, these are quotes from the paper, so don't blame me, okay? Anyway, the most extreme position you could take, I think, is, which I do not agree with, any scale is, change of scale is change of trait. It is true in its effect and the conclusions you may draw, but it just goes a little bit too far for me. The statement that any scale is arbitrary, of course, nobody can argue with that. Even good scales, if you measure, let's say, area of wing, even those such scales, hard measures, what are they? Not interval scales, even better. I forget the word. Even those scales, such scales, are subjected to transformations. So what I consider the pragmatist consolation is that a particular choice of scale will be vindicated by successful prediction that it facilitates. And that is, I think, the key. And that's all there is to it. And what you've got to do is you've got to be explicit about the scale you've used, why you've used it, and present your conclusions conditional on that choice of scale. What else can you do? There is nothing else you can do, because a change of scale will not only change interactions, it will change everything. It may change, it could even change it will change if you use a nonlinear transformation, the contribution of, let's say, additive genes. So there is really, it's not a problem really of G by E, it's a general problem. The quote in the middle, which I will not read to you, is the pragmatist very sharp tongue suddenly, and I think that is in response to Professor Kamen's earlier remark in the paper, where Kamen says that whole undertaking is hopeless because of scale dependency. And this was then dealt with by Lindon. So I'd like to present, I'm here to give a talk about an area of research which I've done almost nothing, except for a recent paper with my student Dolan Molnar. So I'd just like to demonstrate the problem of scale dependency. Now, the figure on the left shows you, for IQ, general IQ, I think, or general intelligence, it shows you the level of A on the x-axis by the environmental variance on the y-axis. And what you see is that the slide is too small to read, perhaps, but as you grow older, the function becomes ascending. When you're young, it's descending. So it goes from descending to flat to ascending as children go older. Each line, conditioning on age. Okay, so I can imagine that somebody could come along and come up with a developmental narrative to explain all this. Okay, but these happen to be samples consisting, it's not one sample, it's many samples. So we can take the 4 to 10-year-old, the descending trend, and we can take a look at the trends within each subsample, and the 5 subsamples shown on the right. Okay, now you may have derived a nice narrative for the figure on the left, but you're screwed, more or less, when you're confronted with the figure on the right. Unless, of course, you think you can have a narrative, you arrive at a narrative to explain this evident heterogeneity. Okay, the quote is from the 2006 paper. This is what is going on. A different pattern of interaction may be detected with different measures of the same construct. Okay, there's one more thing I'd like to say about this paper by Dolan, and that is the possibility of using a common pathway model to model the interactions. This is based on a generalization of the Jinks and Fulker test using marginal maximum likelihood. We fit it in MX, and we use we model heteroscedasticity at the latent level, the latent phenotype, and we model it at the level of the residuals called R1 to R4. Now, we don't interpret the heteroscedasticity at the level of the residuals because we know that it could be due to ceiling or floor effects. We do try to interpret, conditional on our choice of scale, the heteroscedasticity at the latent level. So this may alleviate somewhat the scale dependence, I hope. I don't really believe that. But I like this model. Now, I'm not plugging Dylan's work. I'm merely presenting this slide for 2 reasons. Firstly, it's in the spirit of Lindon's multivariate G by E test. So that is nice. And secondly, it shows you the common pathway model. And that is a marriage of genetic covariance structure modeling and psychometric modeling over which the Reverend Dr. Lindon Eaves presided, because it's present in the 1977 papers. Now, in subsequent years, Lindon has returned to the subject of G by E in a number of very interesting papers. I can't do justice to these papers. I'll just mention that the paper on Monte Carlo approaches is very interesting computationally, but it contains an example of an elaborated G/E model. It's very nice to read because then you actually see what Lindon's on about when he's on about, what exactly he's on about when he's on about G/E interaction. And I'd also like to mention the TDT test because that incorporates the Jinks and Fulker test. So that is this is all very, and the 1984 papers, if you enter into genetic covariance structure, as I did via structural equation modeling, you know, you don't immediately realize that there's Mather and Jinks and books like that and Falconer. Once you start reading those books, the background is revealed, and this is one of those papers that I really enjoyed reading for its biometric modeling. It's a very nice paper, and it's a paper in which apparently there need not be a systematic relationship between means and variances, because in this paper, Lindon doesn't need them. He has two genes in which one can be responsible for a main effect, and the other one can be responsible for an interaction. The second theme of the 77 paper is G/E covariance, and in that paper, firstly, it's interesting to know that it poses a different problem. G/E interaction poses, of course, a conceptual problem and a theoretical problem, but practically it poses a computational problem, how to do it. G/E covariance doesn't do that because you can fit G/E covariance into any path model. It just involves drawing the lines. The problem is, in that situation, is resolution and identification. Now, Lindon, in this 77 paper, discusses three forms of G/E interaction, all theory-based, and that is an important thing because prior to Lindon's work, people would just approach it statistically, thinking of what G/E covariance parameters can I put in my model without reference to a process giving rise to it. And the nice aspect of the 77 paper is that it's based on a conceptual idea of how people will interact, for instance, siblings, or the influence of, mentioned by Hermine, of parents on their children, and how that process, which is comprehensible, gives rise to G/E covariance. Also, Lindon emphasizes the importance of this theoretical-based, process-based approach. Firstly, it's important from a developmental psychological point of view, wherein obviously for children, people become more important than material things. And from a evolutionary point of view, it's important because if I have an influence on the genetic influence on the environment of my siblings, then that is a prerequisite for evolution by group or kin selection. There's one issue within that, is one which dawned on me, or a question that arose, was how, assuming that genes of very small effect can be subject to evolution, how large should this sibling interaction be to actually have an influence on the evolutionary scale? Presumably small. I have to come to a close, I think. I've got 2 minutes, okay. So anyway, the work of Lindon within the realms of G/E covariance based on plausible processes gave rise to a general approach to this problem, which you saw, the upshot of all this you saw in the slides of Hermine. I think, I'm not sure, I realize there's the work of Gregory Carey and others concerning niche picking within and sibling interaction within longitudinal models. I do not think that all possibilities are as yet have been considered. And I'm studying this with my student Unequal, who made this slide for me in the hope that we can arrive at an identified model in which we can demonstrate the absence or presence of phenotype to E transmission over time or A to E transmission over time. Now, we hope to demonstrate that this process is important, or perhaps is not important, in the development of IQ from a differential psychological perspective. I was going to add with this slide, end with this slide, because it gives you an example of a absolutely, to my mind, revolutionary paper. But you've already heard Nick Martin's talk this morning. This had no impression on the community of, I suppose, sociologists or social psychologists. But I'm pretty sure that, I'm glad to hear that it's having an impression now, but I was very surprised when I read this, that in fact, genetic transmission is a much stronger effect of resemblance between children and their offspring in social attitudes concerning, for instance, nudist camps, church authority, pajama parties, or the death penalty. I thought this kind of paper, it involves, in the presence of a phenotypic sort of mating. So it's a very nice example of the kind of modeling that Lindon created, and in its application, I thought it was absolutely terrific. I'm happy to hear that it's starting to make an impression. Anyway, in closing, I'd like to thank Lindon for his inspiring and trailblazing papers, and also his captivating lectures at the various twin courses I attended. And on behalf of my fellow psychologists, my differential psychological colleagues, I'd like to thank Lindon for enlightening the field of psychology.